Nandini ji, are you there?
Yes, sir.
Okay, we will start within a minute. So first of all, thank you, Professor, for acknowledging my request, accepting my invitation to deliver a lecture in this international workshop. I want to let you know that Professor Babak Nakhoda sir is also a chief organizing director of this workshop. And in the collaboration with sir, we are also planning to organize an international conference in June and one international summer school in the collaboration of ABRI, where sir is working, as well as University of Tehran. So thank you so much, sir, for your nice and great efforts. I welcome you in this ninth day of international workshop session. Sir, are you getting my voice, sir? Please confirm.
Yeah.
Yes. Nandini ji, you can start, please.
Thank you so
A
much indeed.
very warm welcome to our distinguished speakers. Good evening, everyone. This side Nandini Sharma. Welcome to day 9 of our international workshop on the topic research paper writing and publication ethics workshop. Before I proceed further, I would like to extend my gratitude to our collaborators and knowledge partners whose support has made this international workshop possible. University of Tehran, UGC Jodhpur, Southern Federal University of Russia, Education University of Hong Kong, China, Agri Biotechnology Research Institute, Iran, ICRISET Hyderabad, ICR Raipur, Chhattisgarh, ICR DCR Puttur, Karnataka, ICR Atari Jabalpur, Madhya Pradesh, Agri Meat Foundation, Bharat, Royal Society of Agriculture, India, Scarlet Wings, India. So it is a pleasure to welcome you all in our ongoing knowledge journey. Each day, we are fortunate to hear from some of the most impactful minds in science and education, and today is no exception. We are honored to have with us Dr. Babak Nakhoda, a globally recognized scientist and a leading expert in molecular plant physiology and crop physiology. He's currently serving as the head of molecular plant physiology at the Agricultural Biotechnology Research Institute of Iran. Dr. Nakhoda's journey is one of the academic brilliance and scientific commitment with a PhD in genetics from University of Philippines, extensive research experience from International Rice Research Institute. His work has significantly advanced our understanding of plant stress physiology and gene expression under salt stress. Having served in this field since 1996, Dr. Nakhoda's research continues to focus on developing climate resilient crops for food security. His dedication has not only led to numerous publications in international journals, but also inspired and mentored countless graduate students along the way. He has also formed some strong collaborations with global institutions like ICRISET and IIMR and works closely with seed companies and food industries, making significant contributions in millet and sorghum breeding, especially in smart foods and sustainable agriculture. Dr. Nakhoda, we are truly delighted to have you with us today. Your insights will undoubtedly inspire and educate us, and we are eager to learn from your invaluable experiences. Let's make today another day towards impactful learning and collaborative growth. Thank you so much, sir. Over to you. The session is yours.
Thank you so much indeed. Thank you, everybody, and good afternoon to each and every one of you. It's a great honor and pleasure for me to participate in this international workshop, and thank you to my esteemed colleague, Dr. Ankur Sharma, for inviting me to be a part of this global movement, actually. And thanks to this very great and very prestigious network that you already made. Great job. Thank you and well done. Just only a small correction on your introduction. Nandin, I'm already joined the systems biology department. I will introduce this. I already left the molecular plant physiology, but it's still at Agricultural Biotechnology Research Institute of Iran. And as Dr. Sharma already asked me to have an introduction about ABRI and its organization charts, its activities, its achievements. Today, I focus in my lecture on introducing Agricultural Biotechnology Research Institute of Iran to you and its objectives, its mission, its achievements, and its outputs. Okay, let me please share my screen with you. So, do you have my screen, sir?
Yes, yes,
Yes, sir.
it's
Yes, sir.
visible.
Okay. So let's start. As I've said, I'm a government employee and I'm working under the Ministry of Agriculture, Iranian Ministry of Agriculture. Now it's being called Agriculture Jihad because it's already merged with another organization working on agricultural aspects and for rural development. So we are under umbrella of ARIO. It is Agricultural Research, Education and Extension Organization. It is the same institute or the same organization as ICAR, Indian Council of Agricultural Research, that you have in India, but a little bit smaller, or just like Embrapa in Brazil. So I'm working at Agricultural Biotechnology Research Institute of Iran. And here you can see the map of Iran and also the ARIO nationwide. So ARIO has different branches, has different research institutions, either focus on some specific crops or some specific subjects in agriculture. So, almost every city of Iran or every province of the state of Iran has a research station and research organization. As you see here, let me close this one and okay. Okay, as you see here, so Iran ARIO has 21 institutes, so research institute under the umbrella of ARIO, and we have 34 research centers and 314 research stations that they are giving different kind of services to the farmers and different stakeholders in agriculture. So among these 21 research institutes, Agricultural Biotechnology Research Institute of Iran is one of the youngest and the most, advanced or pioneering with the state-of-the-art instruments to address agricultural challenges. In this picture, you can see our headquarters, the new building, the new headquarters located in Karaj. We have some other branches in different cities with different mandates. They are located in different parts or different provinces of Iran, but they have national mandates. For example, we have Institute of Animal Biotechnology in Gilan province. We have Institute of Food Biotechnology in Tabriz and Institute of Secondary Metabolites in Isfahan. So all of them are connected with the headquarters and working together. I will discuss about the mandate and their activities and objectives of these satellite institutes later on. When we talk about biotechnology, biotechnology is a very wide spectrum with different color. It's a very colorful area of science and technology. So we have green biotechnology, we have white biotechnology, we have blue biotechnology, we have red biotechnology, and so many other colors for different other purposes. So, but these are the main ones. So through these ones, white biotechnology is for industrial biotechnology. Blue biotechnology is mainly focused on marine biotechnology. Red biotechnology is for medicinal biotechnology and drug discovery. And green biotechnology is agricultural biotechnology. Today, we are focusing on agricultural biotechnology because we are from the agricultural sector, and this is the area of interest. But in the global trend, you can see the biotechnology is one of the most important technologies in the world with a very, very big share of technologies. So even if still artificial intelligence lag behind of biotechnology. So 32% is the share of biotechnology in the context of new technologies. So when you look at the global biotech trade, you can see it's a increasing trade from 2015 to 2024. It is something about $700 billion, and it's been expected to reach to more than $950 billion by 2027. But in agricultural biotechnology, the trend is something this picture at the left side, and in 2024 it was about $75 billion, and it's been expected to reach to $100 billion in 2027. So Iran's current share is $600 million, but it's being expected to reach to $1.8 billion and has a big share in our GDP. So considering this important area of technology, Agricultural Biotechnology Research Institute of Iran, or ABRI, was established in 2000 to develop new agricultural technologies, address main agricultural challenges, conserve natural resources and ecosystems, and also contribute in supplying countries' food security, improve food safety, and promote self-sufficiency in agricultural products. So the mission of ABRI is to develop and utilize modern agricultural technologies to address main agricultural challenges towards sustainable agriculture and food security and farmers' livelihood improvement. And we have a vision to be an innovative, efficient, and effective research institute for development and application of modern technologies in agricultural sectors. So soon this Agricultural Biotechnology Research Institute will be promoted to a new institute for new agricultural technologies, including artificial intelligence. So to be more precise, so our mission is to provide modern technology services to even parent institute, other institutes under the umbrella of ARIO, with the priority of reducing the performance gap, and also to develop and exploit of new technologies with the priority of improving productivity in agricultural ecosystems. So we have strategies in research and innovation. They need to be impactful and target-oriented. This is our approach. And networking with different stakeholders, as well as public and private partnership, is very important for us. Whatever we are doing is not in vacuum. It's in collaboration with some other partners, either national or international collaborators. And we also involve in training and capacity building for next generation researchers and scientists. And also we are working on developing open innovation platforms. This is for transferring and also for commercialization of our research achievements that I will address later on. For this purpose, we are using different technologies. So the technologies that are being used in our institute are including nanotechnology, food biotechnology, microbial biotechnology, secondary metabolites of medicinal plants, bioinformatics, animal biotechnology, genome editing, genetic engineering, genomics and metagenomics, tissue and cell culture, phenomics, remote sensing, and high-throughput phenotyping. And the most important priorities for our research are including these areas, including genetic improvement of field crops, horticultural plants, animal and microorganisms that are being done in different departments and different institutes. So we are using genetic engineering, haploid breeding, and systems biology. and also cell and tissue culture for micropropagation and for production of true to type and virus-free strategic plants. This is also very important, especially for the orchards. Mass production and medicinal plants and secondary metabolites and their controlled environment. This is also very important. This will be addressed later. Development of value-added products from agriculture or industrial waste and residues using beneficial microorganisms in agriculture. We have a very state-of-the-art gene bank for beneficial microorganisms. And also vertical farming under controlled conditions for increasing the efficiency of agricultural inputs and water management, also synthesis and formulation of nanomaterials for agricultural usage in our nanotechnology department, and the development of advanced technologies in tracing, diagnosis, and detection of environmental pollutants and diseases. So these are what we are doing, and in 4 years for our strategic plan, we are planning to reach from these current figures to the expected figures in 2026. For example, in technology transfer contracts from 32 to 73, for joint projects from 30% to 80%, for commercialization support. So we already establishment of 4 innovation centers and 60 technology units that are under construction. So for technical manuals and other services from 20 to 59, for national responsibilities from 4 to 10, this also will be addressed later, and for publishing publications and even for the patents. So patents is not including here, but for publications, almost 150 per year we have, so it's supposed to reach to more than 150 per year. And also about our effectiveness based on the economical effectiveness. Now it's 700 something about it's $190 million per year. So Apple's share in economic effectiveness, but it's supposed to reach $362 million per year. So, and then application of our technologies in farm, it is now at the present is more than 200,000 hectares and supposed to go to more than 300,000 hectares for reducing water consumption from 500,000 cubic meters per year. So it's a very important, especially considering the climate change and global warming and water shortage. So this is very important for us to increase the water efficiency, and increase in production from 300,000 tons to 500,000 tons, and also for the new cultivars to be introduced from 4 cultivars at the moment to at least 12 by 2026, and for technical knowledge from 70 to 112. So as I've already mentioned, the Agricultural Biotechnology Research Institute of Iran established in 2000. Before it was Biochemistry and Plant and Crop Physiology Department under Seed and Plant Improvement Institute, but now it's from 2000 it's become an autonomous research institute with different branches. The Food Biotechnology Institute established in 2004 in Tabriz, in the west-north part of Iran, and also secondary metabolites biotechnology, which working on medicinal plants and secondary metabolites, established also in 2004 in Isfahan. And the latest one is Animal Biotechnology Research Institute, established in 2005 in Rash, in Gilan province, in north part of Iran. So considering the personnel and human resources, we are something about 190 people working at ABRI. So including 7 professors, I mean the faculty members are something about 50, so including 7 professors, 13 associate professors, 28 assistant professors, and 53 experts at working with the in different departments. ABRI has different departments, including microbial biotechnology, molecular plant physiology, systems biology, tissue and cell culture, genetic engineering and biosafety, nanotechnology, that are working together to address the main agricultural problems and to increase the efficiency of agricultural products. Okay. In the genetic engineering and biosafety department, we are working on isolating valuable genes, using plant transformation to improve quality of crops, designing suitable vectors for transformation, performing bioassays and functional analysis of GM plants, and also a study on the biosafety of GM crops. So in this department, we are working on transgenic crops using different approaches to make transgenic crops, either agrobacterium-mediated transformation, bombardment, or ballistic guns. And also we are using the CRISPR, different CRISPR technologies, CRISPR-Cas9, and some other technologies, which being called gene editing technology. So, and also in this department, we established GMO detection lab, which is a reference lab that I will address this issue also later as our national mandate. And in tissue and cell culture department, we are doing mass propagation of pathogen and virus-free plants. So production of haploid and double haploid plants is one of the most important part of this mandate of this department. And also we are using reverse breeding and producing hybrid seeds in vegetables. And you can see some samples of their achievements here. For example, production of F1 hybrid of seeds via haploid systems, especially on bell pepper and cucumber, and also for the mass propagations of the commercial date variety using somatic embryogenesis technique. Another department is microbial biotechnology, which dealing with beneficial microorganisms in agriculture for different purposes. So they are working on production of products at industrial scale, also production of biological products, for example, pesticides, fertilizers, and food. Also preparation of genomic libraries, identification and preservation of agricultural microorganisms. So they are also working on production of different pre, pro, and symbiotics for poultry, for animal husbandry, for fisheries, and also using beneficial microorganisms for waste management, for management of agricultural waste and residues to produce biogas and biofuel. And in molecular plant physiology, we are working on a study physiological responses of crop plants to environmental stresses, either biotic and or abiotic stresses, investigation of molecular mechanisms responsible for tolerance to biotic and abiotic stresses, development of new methods for physiological studies in crop plants, and also study new alternative and climate resilient crops. So you can see some of the samples that will be addressed later. You can see here our cool season grasses at the left side, and also our work on summer crops, climate resilient summer crops, including different kinds of sorghum and millets, thanks to India, ICRISAT and IIMR, and also on halophytic and xerophytic species like Salicornia, Sueda, Salsola, and so on. And in my new department, which being called systems biology, it was before functional genomics, but now it's already changed to systems biology department. So in this department, we are working on molecular breeding, molecular assisted selection, molecular assisted backcrossing, and molecular assisted breeding, including sequencing the genomic structures of crop, insects, and microorganisms. Also determining the genetic originality of plants, animals, and pathogens. This is also very important for some other institutions who are going to release new cultivars, new varieties. Identifying the candidate genes involved in environmental stresses. This is also in collaboration with some other departments and institutes, including molecular plant physiology and some other universities, and developing the pipeline system for multivariable data analysis. In this department, we are also working on isolation and screening enzymes for different purposes to be used also in agriculture and in industry. So these are the wide spectrum of different enzymes with different purposes and different applications. Some of these projects was including molecular modification of almonds and also production of meat and breeding sheep in collaboration with the private sector. And the new department is our department is a nanotechnology department, which working on synthesizing nanomaterials with potential application in agriculture or nano-agro products, design of nano-biosensors, improving the efficiency of pesticides and chemical fertilizers, and reducing agricultural waste products. And you can see some of the samples of their work here for nano remediation, for slow release fertilizers in collaboration with the private sector, also for coating producing coating products for increasing the shelf life based on the nano products, and design of colorimetric kits for different purposes, especially for the for fisheries to measure the ammonia in the water. It is very important also. But regarding the satellite institutes that we have, first we go to Food Biotechnology Institute located in the northwest of Iran in Tabriz. So this department is mainly focused on production of high-value compounds from microorganisms and agricultural waste, and also developing micropropagation of native horticultural plants. They are also working on identifying and isolating beneficial microorganisms associated with food industry and producing secondary metabolites for food and cosmetic products. And you can see some samples of their work here. For example, production of fermented milk, which you can conserve it for 6 months without refrigerator, and production of cosmetics from microalgae. So these are the samples produced, the shampoo and some other products produced from different algae, including Donaliella from Urmia Lake that can grow in very saline water. And this one at the right side is our photobioreactors for algae, development of microalgal cultivation systems. It's a very unique and a state-of-the-art system that can use the CO2 from the generators, power generators, and also the solar energy from the sun to produce biomass of algae, and also to be used either for animal feed or for making some extracting some value added products from the algae. And in Isfahan, in Secondary Metabolite Biotechnology Institute, we are working on developing protocols for mass propagation of selected medicinal plants, molecular breeding and genetic engineering of various medicinal plant species, and also optimizing secondary metabolite production in bioreactors. You can see some of these samples here. Instead to go to the field and cover 1 hectare, 2 hectare to produce the biomass and then go for the extraction of the bioactive materials, bioactive compounds from the biomass. You can make it here in the lab, hassle-free, no need for the land, no need for that much of water, and you can produce a huge amount of secondary metabolites in these homemade bioreactors and very easily, and it's very environmental friendly also. So you can see it here, and also at the right side you can see our homemade plastic bag bioreactors that you can produce different secondary metabolites from different medicinal plants in these bioreactors, hassle-free and no need to the land and too much water. So it's good under climate change conditions. Okay, and in Animal Biotechnology Institute in Rash, in Gilan province in north part of Iran, we are working on breeding and genomic selection of livestock, poultry, and insect using omics and genome sequencing technologies. This department also working with our systems biology department in Karaj headquarters, genetic engineering of livestock, poultry, and aquatic animals. It is also aquatic animals, fish are very important. So different kind of fish, also estrogen fish for caviar extraction, optimizing production and breeding of livestock, poultry, fish, and industrial insects, and producing feed supplements for livestock, poultry, and fish to increase the efficiency. We are also doing some genetic analysis for the high value animals like horses for the genetic purity and genetic identification of different horses, because this is a big industry and big economy. So at ABRI we have different priorities. So for propagation and preparation of virus free seeds, we have 10 strategic horticultural crops. For hybrid seeds, we are now focusing on pepper and cucumber. On molecular breeding, we are working on different kinds of animals like sheep, Aryan line, which is a chicken, and also estrogen and trout fish. And we are also, for molecular breeding, we are also working on plants like millet, sorghum, safflower, chickpea, grasses. So these are in the next part, development of alternative climate resilient plants. For this one, we are using different technologies, including molecular breeding and molecular assisted selection. Also, technological production of bio and nanofertilizer and pesticides. 5 important export products are being addressed. And also for conservation of agricultural waste and residues, we are working on compost and humic acid. This is very important also for the carbon sequestration and also reducing the emission, and also for the waste management and for taking care of the environment. They are very environmental friendly technologies. And processing and standardization of medicinal plants. So 3 strategic products of medicinal plants are being under research. Technological production of food supplements, including macroalgae, of unconventional water and probiotics, production of transgenic products, including cotton, soy, and canola, and also emerging technologies, including artificial intelligence and artificial biology. But as I've already said, we are not working in a vacuum. So we're considering biotechnology in the context of systems biology. So we are addressing different parts of a system to help the stakeholders to get more benefit from their farm, from their crop, from their animals, from their fish, or even from their microorganisms. So in the area of agriculture, for the field crops, we are working in the field and in different under different conditions. For example, this one is under normal conditions or under saline conditions. We are measuring different criteria, different characteristics, different traits under different environmental conditions to address the main mechanisms and the main challenges that can lead us to superior varieties more tolerant to harsh environment, including global warming, climate change, water shortage, heat, drought, and salinity, and even alkalinity, or even for the for biotic stresses. We are also doing so for the pathogens to produce pathogen-free or pathogen-resistant or insect-resistant crops. So all these works are being conducted in collaboration with different mother institutes, different departments under the RU, and in different research stations, in different agricultural centers. So these are also some works in collaboration with the University of Kerman as the biggest university in the southeast of Iran. So hopefully we'll join this network shortly. So these are some of our work in collaboration with the Kerman University and their Saline environment. And these are my colleagues, Professor Mohammadi-Nejad, who's the Director General of the Plant Production Research Institute under University of Kerman. And these are our lysimeters. So these are also our work. So we are working on the field and also in the under control conditions. So we are also screening our materials. We are making the mapping populations, doing parental survey, making mapping populations, and screening the mapping populations under different environmental conditions. This is under salinity stress. We are also doing under drought or some other stresses. And these are our PhD students working working at ABRI, but they are from different universities. We also have our own PhD students. So these are to understand the mechanisms that are responsible to make one crop sensitive or one crop tolerant to any specific environmental factor. For example, here an example about salinity, and we are measuring different traits for phenotyping analysis, including morphological, agronomical, physiological, biochemical characteristics that we call it phenotyping. So these phenotyping are being made mainly in molecular physiology department, and then we can go for the genotyping, and then in systems, in Department of Systems Biology, and then the data from phenotyping and the data from genotyping will be combined in our bioinformatics department for understanding the chromosome loci or the QTS or the genes or the markers that are involved in their for example, response of any specific crop, any specific variety to any specific environmental stresses. And this is very important for us. So here we can understand the mechanisms that involve intolerance or insensitivity of a crop or a variety to for example, to drought or salinity. And then you can go for gene discovery, or you can find the QTS or chromosome loci that they are responsible for these responses. And then you can go for the process to pyramid these genes or these QTS in one single variety and bring a new variety for the farmers that can tolerate harsh environments in those areas that are hotspot for heat, for salinity, for drought stresses. Okay. To have a summary of research projects and outcomes and impacts of ABRI. So, so far we have more than 40 technology transfer agreements, and 6 of them already commercialized technologies. These are the impact of our institute. We have more than 70 invention disclosure and know-how. Also more than 8 technical instructions and more than 700 final reports from our projects, and more than 3,000 articles already published by our faculty members. And this is for your information that ABRI is the first rank in publication and also impact among those 21 research institutions under RU. Although it is the youngest, but it is the first rank in science and technology. So about the most important achievements, these are some of examples. We are working on management of agricultural threats, and this is a very important achievement for us. Management of witch's broom disease of Lyme for the first time in the world. So as you see here in the green part of Iranian map, so these areas, more than 47,000 hectares, are covered with the lime trees, and they are being threatened by this witch's broom disease. And ABRI was the institute who was able to collaborate with different research institutions, including Plant Protection Research Institute, to address this challenge and find a way and already manage and control this disease for the first time in the world. So, and we also have already established state-of-the-art transgenic greenhouses, BL2 and BL3 greenhouses for transgenic crops. So you can come here and work on your own transgenic crops here if you would like to work on genetic engineering using different approaches. And this is another example, establishment of virus-free potato mini-tuber seed technology, that it was also a big milestone, big achievements for the country, because now country becomes self-sufficient in importing potato seeds. So all of them are being produced inside the country. So in cooperation with 25 companies. So, and then we have self-sufficient in potato seeds, 500,000 tons per year. So, and then we were able to create job for 220 people working with those 25 companies, and its economical impact is at least $125 million per year. And from our microbial biotechnology research department, production of a specific probiotics, prebiotics, or even symbiotics for poultry, not only for poultry, even for human, for animal, so for even and also for the fisheries. So these are some samples. Total production is 500 tons per year. The value of products is $300,000 US, and its economical impacts is 17 million US per year, because it caused increasing the efficiency of the animals and decrease in feed consumption and feed waste. And another one is mass production of Malling-Merton apple rootstock using tissue culture technology, and its economic impact is estimated about 50 million US per year. Average yield increase per hectare was 20 tons per hectare, and job creation in tissue culture for something about 170 people. Another one is industrial production of trichoderma-based plant biotics. This one already transferred to the private sector, and with in collaboration with the private sector, they are using these commercialized trichoderma products, which cover coverage of 20,000 hectares per year, and yield increase in different field crops and horticultural plants. At least something about 30 to 60% already reported. And it can be used as biocontrol efficiency 70 to 100% as the biocontrol efficiency. So it's a very, very beneficial microorganisms, trichoderma. I'm sure you also have in India. And another milestone is tissue culture propagation and virus-free plant of sugarcane cultivars. Sugarcane is very important in Iranian agriculture, because in Khuzestan province, located in the south west part of Iran, in the borderline with Kuwait and Iraq, we have more than 100,000 hectares under different cultivars of sugarcane cultivation in 10 sugarcane agro-industries. This is a huge, huge area under sugarcane cultivation. And we work with sugarcane agro-industry and byproducts company on establishment of primary sources of sugarcane nurseries for selected tissue culture cultivars, which preventing 1 to 3% sucrose loss due to cultivar mixing. So it is something about $200 per hectare. So multiply it by 100,000 hectare. So you can, it's a huge number. So another achievement was enriched biocompost production from sugar cane residues, either bagasse or stalks. We have something about 4 million tons per year, and it's agricultural waste. So you cannot use it for directly, I mean, as to be used as compost or to be used as a animal feed. So but our colleagues from Department of Microbial Biotechnology work on it for many years, something about 7 years working with the sugarcane agro-industries. So the total production of enriched biocompost now is more than 10,000 tons per year with the economical value of $2 million per year. So it's become a huge industry for sugarcane agro-industry. Another achievement, which is very new, using reverse biotechnology or reverse breeding, is production of the hybrid seeds for pepper and cucumber. So 2 F1 hybrids already released. So this is a hybrid technology using the macrospore culture and haploid breeding technologies in our tissue and cell culture department. Another one on sheep breeding, molecular genetic production of new strains of Iranian sheep with industrial breeding capabilities. 2 genes already transferred to the Iranian native sheep, and now they have more breeds, and also they have more muscle. You know, the ratio of the muscle or meat to the fat is already increased. So it caused the efficiency and better income for the sheep keepers. And this is some samples of my own work in collaboration with Kerman University and some other mother institutes, including Seed and Plant Research Institute, on production of alternative and climate resilient grasses, including summer grasses, including different kinds of sorghum and millet, in collaboration with ICRISAT, IAMR, and Indian Institute, and in collaboration with Nebraska Lincoln in USA, and also on cold season forage grasses at the right side, which are perennial grasses that can use the green water during rainwater or snow during the winter and autumn. Because here in Iran, we have 4 seasons. So if you have a crop that can use rainwater, is very, very good for, during the climate change. They also very, very, very effective in carbon sequestration. So considering the different agricultural challenges that we have, the breeders facing so many troubles, so many challenges. So poor breeders have to produce more with less. So with less water, with less human resources, with less money, with less arable land, agriculture has to produce more food for the increasing population to ensure food security. So it's a big challenge. You can see the figures here, and it's really big challenge for all the governments, for all the countries, especially for India. It's a hot spot for so many environmental stresses, and also has a huge population. That's why we start to study climate resilient crops in the context of climate smart agriculture and low input sustainable agriculture, or LISA. So these crops, as I've already said, including the summer crops, including different kinds of millets, including sorghum, and also cool season forage grasses as winter crops or perennial crops. So these grasses are good to be used in pasture as silage, also for the rangelands. They are also good to keep the soil health and erosion, and also they are good for water resources because they help to penetrate the water into the soil. So, and then they can enrich the groundwater resources. And they are also very good to maintain and increase the biodiversity. You can see here our field. So it was a bare land, but now you can see so many birds, so many insects are moving around. And these are the different genus, different genres, and different species of our grasses that we are working. So they are including 16 different species of different grasses. And also our summer crops, as I've already mentioned, including different kinds of millet and sorghum. You can see here proso millet and so foxtail millet. We also have the pear millet and sorghum originated from India. We did the evaluation for Iranian germplasm in different cities, in Yazd, in the central part of Iran, in Karaj, in northern part of Iran. And also we had a good collaboration with Professor Doveykat from Nebraska Lincoln. We already received some materials from him from Nebraska Lincoln, especially sweet sorghum materials. And these are our a small millet farm, including foxtail and proso millet germplasm. We started some collaboration from 2015 with ICRISAT, thanks to Dr. Gupta and Dr. Ashok Kumar, and also Dr. Wright. So they are very, very collaborative, and we were able to visit their farm, and we inspired with their very state-of-the-art, very, very fantastic work on different kinds of sorghum and millets. And through this collaboration, we were able to start negotiation between RIO and ICRISAT, and the memorandum of understanding already signed, and the high-ranking delegation of ICRISAT visited Iran in 2018, and very valuable breeding materials we already received from ICRISAT that will lead in near future to new cultivars, new varieties and hybrids, and also OP varieties of different kinds of pear millet and sorghum in Iran for the first time. So, and these are some samples of the performance of the ICRISAT materials in our locations, in our fields. So, and this is for sorghum also, and you can see the sweet sorghum with 5-meter long and 23% of bricks. Sorghum can perform very well in Iran also, and it has great potential. We are working also in introducing an extension of these new crops as climate resilient alternative crops to secure and to supply food security under climate conditions. So this is very important for us to, familiarize and also to aware farmers and stakeholders about the new crops and the potential of new crops, especially millet and sorghum under climate change conditions, not to go only for maize or alfalfa, which they need a huge amount of water. So we already signed some contracts with the private sector to introduce new cultivars of pear millet and sorghum. It was for the OP varieties, and the hybrid varieties will come later. And also establishment of the first natural stevia sweetener factory. So it's already established in collaboration with the private sector. And also supply microalgae for industry, as I already mentioned in our Food Biotechnology Research Institute in Tabriz. These are from Donaliella and Spirulina microalgae that are being used also for feed, for food, also for cosmetics. And we also have some reference labs for imported genetically modified organisms. Every year, more than 12 million tons of imported products are carefully evaluated if they are either transgenic or non-transgenic. If they are transgenic, it's okay, but they need to be approved by the Food and Drug Administration in Iran. So we also have reference lab for nanomaterials, nanomaterial-based products. We have a reference lab for fraud detection of herbal medicines, and also we are a lab partner of the veterinary organization. Our impact, as I already mentioned, for annual economic effectiveness is more than $190 million per year. Usage in the field and garden is more than 200,000 hectares, and increased production and reducing waste is 300,000 tons. And regarding the know-how and technological products is something about 70 at the moment. And for the Innovation Growth Center in collaboration with Park, now we have 4, but they will be increased soon. So for the core and technology unit and companies, 20 at present we have. So to summarize this one, so this is the picture. So 190 million US dollar per year. It's the big impact of ABRI in Iranian economy, and also 200,000 hectares of cultivated land per year under our release cultivars. But the most important activities in progress, so we are still working on a study introducing an extension, new alternative and climate resilient crops, and millet and sorghum program is one of the most important programs that we have here in collaboration with some other related institutes and universities. So it's a very comprehensive research program starting from germplasm exchange, germplasm evaluation, to in producing elite lines as parental lines for hybrid production, and also for OP variety production using different technologies, using different techniques in molecular phenotyping, remote sensing, phenomics, genomics, marker-assisted selection, marker-assisted breeding, to variety release and introducing new cultivars to the farmers. And another pipeline is improvement and promotion of Aryan strain, which is an Iranian strain. So hopefully in very near future, we have Aryan Plus using, again, different technologies, especially genomics technologies and SNP cheats, and also bioinformatics and bio data. And we also have some projects in collaboration with the private sector, production of biological fertilizer and pesticides. These are all in collaboration with the private sectors. You can see the name of the companies here. And also production of dairy starters. Also, these are being done with our food research institute, Food Biotechnology Research Institute, and also with the private sector. And these are very, very famous brands in Iran for dairy products. Another one is production of nanofertilizer, including nanofertilizer and straw release fertilizers using nanotechnology. This is also a joint collaboration with the private sector, and being funded by the private sector. And another funded project is for genome sequencing and also sequencing editing. The first one is for barberry genome sequencing. It's being done in collaboration with the South Khorasan Governorate, so with the Agricultural Research, Education and Extension Organization and some other partners. And we are also working in Isfahan on vertical farming for increasing the efficiency of of the land and water and their control conditions for food security. This is also very important. It's a trend actually in the world. You can see it in Netherlands, in Japan, in even in US and some in Europe, in some other countries. I'm sure you also working on this technology to produce even vegetables or even croplands in a very, very dense conditions and environment with less water, with less soil. And again, we are also focusing on the development and promotion of a specialized innovation center for new agricultural technologies. This is very important to transfer the technology, to transfer the research achievements from the research, from the lab, from the greenhouse to the stakeholders. So for this purpose, we already established Innovation Center for Food Industries in Tabriz, in collaboration with our Food Biotechnology Research Institute, and also in Karaj in headquarters at the right side, we already established Innovation Center of New Agricultural Technologies, which cover an area something about 800,000 square meter, and some SMEs, startups, even incubators, and accelerators are already located in this innovation center. As I've already mentioned, we have some national and international responsibilities. For the national responsibilities, we have National Reference Lab that I already mentioned about GMO detection, and also reference lab for nanomaterials, and also the collaborator of veterinary organization. And we are the national focal point of biosafety because we are working on the GM crops. So that's why we are also focal point of we are the national focal point of biosafety in different international meetings and gatherings or forums. And at the international level also, ABRI serves as the international focal point of EcoAvian ECO Agricultural Biotechnology Network, that we are doing some training courses and also some workshops for the ECO member states, including 10 countries in the ECO region, including Turkey, Afghanistan, Pakistan, Iran, Kazakhstan, Uzbekistan, Turkmenistan, and some other countries from CIS region. So all these works that are being done, all these projects, all these achievements contribute to the UN Sustainable Development Goals that you can see some samples here. So at least 12 goals out of 17 goals are being addressed by our work at ABRI. And it was an introduction about our work at ABRI and its achievements, but since this international workshop is mainly focused on the publications, I would like also to mention about publications and patents. So there is a difference between these two. So if you want to go for patents, so you are not allowed to do the publication before that. So even sometimes when you are working for an agency, you are for example, you're an employee working in Google, working somewhere else, or working in research institute, working for a seed company in agriculture. Maybe you are not allowed to publish your data freely. So sometimes it is trade secret, sometimes it is need to be patented. So that's when you cannot publish it. But if you want to publish your paper, you need to publish your research work, like what we are doing at ABRI. As I've already mentioned, we are publishing more than 150 scientific papers every year. So you can go to this service from Taylor Francis, how to publish your research, a step-by-step guide to getting published. So this is very useful for all of you who want to publish your paper. As you better know, and I'm sure that my esteemed colleagues already addressed these issues, that the publications need to be new, innovative, clear, original, and need to adding new things to human knowledge. This is very important. And it must have a clear message or instructions for the readers. So be careful with this one, and I wish you all the best in your research activities and publication. Thank you so much indeed. Thank you, Dr. Sharma.
Thank you so much, sir. Dear Nandini, are you there?
Yes, sir.
Yes, please
So
continue.
everyone, if someone is having any doubts, any queries, the chat box is
Any
open.
participants has any query related to teaching, extension, and research, and study in every study in abroad, especially study in Iran, है ना? He or she can discuss. ठीक है? If you have any query in particular topic like research paper writing and publication ethics, patent publication, copyright publication, sir has
which is the
vast knowledge in such type of academic
Sir,
things you can discuss.
sir, which is the
Yeah,
best
Dr.
reference
Sharma,
to
as I've already mentioned, we are working on capacity building and training. So that's why ABRI is open for any scholars to come here. All scholars are welcome to ABRI or even ARIO in different institutions under ARIO. We can manage it. So, and then if they want to go, I mean, the graduate students can go for this, especially PhD students. They can come here. They are most welcome for their sabbatical, for internship, and also the Iranian journals are open for any accepting research papers, original research papers from abroad. So especially we have, as I've already mentioned, we have agricultural biotechnology journal in the University of Kerman accepting English papers, and also we have crop production journal in Gorgan University. They are also accepting. So different journals in Iran accepting English, French, even German papers.
Excuse me, sir.
Yep.
Sir, can you please explain me about some of the internship opportunities?
Okay, so for the internship, you may apply through ARIO international offices. Okay, so and then it depends on the area of your interest or what you are doing and your capabilities and skills. And then after that, they will. I mean, ARIO International Affairs will do so, and then they dispatch different requests to different research institutes, so related research institute. For example, as I've already mentioned, at ABRI, we have different departments. This request is related to any departments or related to ABRI, they send it to ABRI. If it's related, for example, to animal sciences, so it will go to that institute. If it is related to dryland agriculture, will go to that dryland agriculture. So this is very routine, and there is no problem for that. For your information, we had some trainees from Uzbekistan, from Brazil, and also from Venezuela recently. So
Sir,
they
we have
came.
to apply through website only.
They can apply through website. For those, you can go to ARIO. If you know the institute, the specific institute, you can apply directly to that institute. Otherwise, you can write to ARIO International Affairs, and then they will dispatch, and they will send the request to the related institute.
Thank you so much, sir, for this wonderful
sir,
session.
sir, I'm a biotechnologist. Sir, how I can use CRISPR-Cas9 in soybean mosaic virus? Sir, it's a big problem.
Pardon, I didn't get your message.
Sir, I'm a biotechnologist and I'm working on genetic engineering and genome editing.
Okay.
So how I can use the CRISPR-Cas9 in soybean mosaic virus, sir? Can you advise me some tips, sir?
Yeah. Actually, now we are using the CRISPR-Cas9 in our genetic engineering department. So one of my colleagues, you can refer to our website. So our English, we have an English website, and then you can go from home to the departments, and from departments you just go to the Department of Genetic Engineering, and then in Department of Genetic Engineering we have different faculty members working on genetic engineering using different approaches. So for the CRISPR-Cas9, I think the best one is Dr. Mohsenpour, Motahhare Mohsenpour. So she's very good in
Doctor?
CRISPR-Cas9.
So can you repeat it, doctor?
Motahhare Mohsenpour. I will write it in chat here.
Sir, much obliged. Sir, which university?
From ABRI.
Okay, sir.
From ABRI Genetic Engineering Research Department. Okay. But for your information, ABRI is not the only institute who's working on genetic engineering. We are under the Ministry of Agriculture, okay? But we have another institute, is National Institute for Genetic Engineering and Biotechnology. So it's being called NIGEB. So like this, National Institute for Genetic Engineering and Biotechnology. NIGEB is under the Ministry of Science, MSRT, Ministry of Research and Science, Research and Technology. So in this NIGEB, they are also working on CRISPR, genetic engineering using different approaches. So, okay. So they are using also genome editing, different genome editing methods, not only for the agricultural, crops or animals, but also for drug discovery, for different kinds of, for the wider spectrum of biotechnology, not only agricultural biotechnology. So you can also search it, and then you can go to their website, and then you can find the right person. Otherwise, I can you just send an email to me, or through Dr. Sharma, I can introduce you to the right person.
Sir, your email can you please provide?
Yeah.
So can you guide
Is
me
there
whether I should
anything else? Please.
sir, whether I should do PhD in biotechnology in India or in abroad, sir?
Again?
You wish to
Whether I
do
should
PhD in biotechnology from, Malab, he's asking, he's interested to do PhD in your country, sir. Is there any opportunity in the specialization of agriculture
Sure.
biotechnology?
Yeah. Let me make it clear for all of you. So the ministry who is in charge of higher education for BS, MS, PhD, graduate, undergraduate is Ministry of Science, Research and Technology. Okay, and all the universities are under the umbrella of Ministry of Science, Research and Technology, except for medicine. So medicine is different. So the medicine is under the Ministry of Health and Medicine Education, but the other majors, including the agriculture and some other, other majors related to science and technology, they are under the Ministry of Science, Research and Technology. And all the students who are going to come to Iran, for example, from BS to MS or from MS to PhD, they need to apply through Ministry of Science, Research and Technology. So, and then if you already admitted to an Iranian university, for example, Tehran University, Mashhad University, Kerman University, you can do your coursework there, and then you can apply for your research either in the university or in the research institute, like here, like ABRI. So it is very routine. like me. I did my PhD in the Philippines, in the University of the Philippines, Los Baños, but I did my research at IRI, International Rice Research Institute. So you can do it as you wish. But we at the Ministry of Agriculture, we cannot accept the students directly. It should be through the Ministry of Science, Research and Technology, because they are the one who will give you the certificate, the PhD certificate.
Sir, your
Okay.
mail ID, sir.
I will send you my email ID here.
Yes, sir.
Anything else, please?
Okay.
I think no one in the chat box or no one in
Whether,
comment section. Nandini, please
Sir,
give
whether
concluding
it's
remarks.
On behalf of all of us here, I would like to express a sincere thank you to Dr. Babak for being with us today, listening everything your work with full of dedication, hard work, ethical research was truly inspiring. Your words were just not informative, but truly inspiring. The way you shared your journey, work with such clarity and warmth touched all of us. We are grateful for your presence and for the knowledge you passed on to us. Wishing you continued success in all the amazing work you do. Thank you once again, and thank you everyone for being with us today.
Thank you so much.
And also thank you from my side, sir. You are very humble and polite person. Whenever I approached you regarding any work related to teaching, extension, research, academic activities, or collaboration with Irani universities, you always be there, sir. Really.
Thank you.
I
Thank you
appreciate
so
I
much, sir.
appreciate his humbleness, sir, definitely.
Thank you so
And
much,
looking
sir.
forward your support in our upcoming international conference with your esteemed organization, summer school, and 3-month skill development program.
Sure, definitely. Because I know most of these guys, they will be the next generation of agricultural scientists, university professors. And I wish all of you all the best. And we, including me, Dr. Sharma, and all others in the research institute and
Sir,
in the university, working so hard
sir, one question, sir.
to transfer some of our, our experiences and knowledge to you. And hopefully it will be useful for you and for your future.
Sir, one question, sir.
Yeah.
Sir, whether we should do PhD in an institute or a university, sir, which will be better?
Panda, please. He's about to close it.
As I already said, it should be in the university, because in a research institute, they are not allowed to give you PhD certificate. You can do your research. You can come as a sabbatical and do your research in the research institute. Otherwise, you should go to the university.
In the research.
Yes, Panda, yes.
Okay, I think query resolved. All participants, I want to let you know that you will get your workshop certificate duly signed by the sir signature, because sir is the chief organizing director in this workshop. So behave in a very polite or humbled way. Okay.
Thank you so much. Thank you. Very
Thank
kind
you
of you.
so much, sir, for joining.
Thank you,
Thank you so much for
everybody.
joining
all
us.
participants. Thank you.
Okay. Good night, sir.
Bye-bye.
Good night. Okay.
Good night. Bye-bye.
Bye-bye.